Okay, we're going to get started. The online people can see there's cameras around and the speaker in the middle. Let's just start with the acknowledgement of country. Are you gonna do one? No. So the University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contributions to Australian and global society. And today I'm very happy to have Drew, who did his PhD at the same place as me, but we just missed each other. But we have reunited for real this time here in Brisbane. He works just across the river at CSIRO. So people at UQ who want to get to know Drew, it's very easy. So he did his undergrad in America at Rutgers, his PhD, Berkeley. Then he worked for the TNC, the Nature Conservancy, before coming here and starting a position. He'll be here at least 4 years for this position. His work, he's a landscape geneticist. Yeah. And his work today has been featured all across the different media outlets that cover science. And I'm very excited to hear about it. So thank you.
Cool, thank you. Morning, everyone. Thanks for having me. I'm excited to be able to share this with people because it's been a long time coming. And I want to just encourage people, two things actually. One is I tweaked my neck a couple of days ago. I think I slept through it. So if I look like I'm moving like a robot, and I'm not a robot. And the other is I highly encourage people to bring up not just kind of things they're interested in, but also kind of critiques and kind of counterpoints, or I'm not sure I buy that, because I've been really eager to kind of dive into kind of the weak points here that I think would be fun to follow in the future. But so what I'm going to talk about today, as you can see here, is global phenology maps and how they can reveal the drivers and effects of seasonal asynchrony, which I'll define in more detail. This is work that came out of, this is the final chapters in the PhD dissertation, so it came out of years of work starting at University of California, Berkeley, and continued through when I was working at TNC and CSIRO. Co-authors, Sarah and Lauren, were undergraduate mentees of mine for the years, and Ian Wong was my advisor. And so the inspiration for this work was an interest in seasonal asynchrony from the point of view primarily of evolution, and a particular hypothesis, which comes out of a small paper from kind of a strange college or university-hosted journal that I bumped into years ago when I was in the field, and it kind of became a brainworm and it never got out of my head. So it starts like this. It says at high latitudes, the predominant forms of seasonality are day length and temperature. And these are mainly controlled by orbital mechanics. So just the angle of the Earth relative to its orbit around the Sun. Which means that these forms of seasonality generally should be broadly synchronized across landscapes, and not just landscapes really, but broad regions, longitudinally. And this is from that original paper, where they, in the pink boxes here, map spatial asynchrony of a few different climatic forms of seasonality. So solar radiation on the top, temperature, precipitation going on. And they found that generally, precip being the main exception here, generally that is the case. There's sort of, brighter, hotter colors here are more asynchronous across space in terms of the timing of their seasonality in these variables. And what this means is that populations of a species should generally breed at the same time of the year across the landscape. They don't need to be separated by mountain range, but in this cartoon example, you've got a species of birds that are all in breeding plumage, regardless of where they are on the landscape. So their mojo meter on the y-axis here, this could be some sort of measure of behavior or hormones or what have you, is basically synced up, right? And the given day of the year, they're at the same point in their sort of annual mojo cycle. But what that means is that chance migration between these populations would lead to reciprocal breeding behavior and love and gene flow and babies. Where this becomes interesting is at low latitudes. In these authors' original hypothesis, in which they say that the predominant forms of seasonality at lower latitudes are precipitation, cloud, and light as attenuated by cloud cover. And that these are mainly controlled by atmospheric circulation and landform, and really the interaction of which are complex topoclimatic dynamics. And this means that seasons could potentially be asynchronous across landscapes at lower latitudes. And again, they in their results, they found evidence. And then, if we're looking at the same cartoon species, but now this is a, going back to boreal species, what this means is that populations could be breeding at different times of the year. So when these guys are breeding, these are not, and vice versa, and they're between shall meet, which means that the chance migrant between populations wants to breed, gets that, and they're the voters. And the most important bit here is that this would lead to evolutionary isolation between these populations, and given enough time, they hypothesized could lead to speciation. So to sum this up, the term that we assigned to this in our paper is asynchrony by allopatry, which is the only thing I'm going to read point for point from the slide. I promise, but this is the crucial point that everything else in the study sort of hinges on. The idea is that ecological processes in different locations can be decoupled not only by the physical distance between those locations, so that's the allopatry part, but also at the temporal displacement between their asynchronous seasonal and phenological cycles. That's that allopatry. And the grand picture in this original study was, and the grand question was really, could this help explain the latitudinal diversity gradient? Is it possible that speciation rates are higher in tropical mountains because of this, and that that's why they serve as species pumps, and that's why there's so many more species there than elsewhere? And this is the contemporaneous latitudinal diversity gradient in mammals from Manning et al. 2014. And so this was fascinating to me, and I began to think of this in the context of the global geography of the climatic controls on seasonality, on phenology. And there's a bunch of kind of pieces here I'll throw at you just as sort of, to give you a flavor for the original inspiration for where we took off from. So Nemani in 2003, based on a mechanistic model, mapped the three main controls on plant productivity and therefore, resource production basically in ecosystems globally as temperature, radiation, and water, and find quite complex geography of the controls on these things. And there are regions where there's pretty quick turnover in space between things like predominantly water control and predominantly radiation and temperature control. And when you layer on top of that the global monsoon system, especially in some of those same regions, for example, where there are places, the blue here, anything that's not blue or yellow can get rain throughout the year. Blue and yellow are monsoon regions, and the blue here are places where the boreal winter, so northern hemisphere winter, it's predominant rainy season. Yellow, it's the opposite, the boreal summer. And there are areas where there's a pretty sharp turnover in space between these. And you layer on top of that places around the world where the warmer colors here, or the darker colors, I guess I should say, are places where there's stronger signal of bimodal precipitation throughout the year. So even within regions that are predominantly water controlled, there are regions where there could be 1 or 2 rainy seasons throughout the year. And even things like cloud cover, mapped here by Wilson et al. In 2016 in PLOS, has a huge variability globally in terms of the seasonality. The color on this wheel is the month of peak cloudiness, and the saturation, so further out in the wheel, is a stronger signal of seasonality. And you get really complex patterns even at local scale. So it's not that strong, but it's definitely pronounced. Even between neighboring valleys within the Andes in northern Colombia, for example, you can get peaks in cloud cover that are nearly 6 months out of sync, as if they were in opposite hemispheres. So that's the background at which we walked into this study. And it set up sort of a series of research questions. And the first was simply, how does seasonal phenology vary globally? So we know a whole lot about the global variation in climate, like we just said, and the potential controls on phenology. But it turns out that mapping phenology globally is not only had not really been done, but was incredibly there's very little information on the spatial variation in phenology at the global scale. The field that does this primarily is called land surface phenology, and it's essentially using remote sensing data to map seasonal changes in surface vegetation. And again, assuming that surface vegetation is a proxy for phenology kind of across species and at the ecosystem scale. And there's a major question mark, right? And that's sort of the strong assumption I made going into this work, is that if we can map phenology from space, should that be a strong predictor of individual species phenology, at least for some species? But there are a lot of limitations to doing this, and the two main ones are one, that there's really just been a lack of generalized global model or methods for mapping phenology. So a lot of methods are applied to particular agricultural systems or particular regions, but there are plenty of places where they fall down. Here's an example of the MODIS. So MODIS is a remote sensing sensor, right? This is the MODIS program's own land surface phenology product, and all of these red areas are areas where they have no data because the methods that they use for fitting the peak of the growing season simply don't work, because there's not a simple development season, or there's very little signal to noise ratio in arid regions. And the other thing is that a lot of the remote sensing metrics that were used to do this had fairly low sensitivity in some biomes, a lot of the same biomes, arid regions and the wet tropics especially, leading to things like this. Papers, I still find this funny, a pair of papers titled Amazon Forest did green up during the 2005 drought, they did not green up during the 2005 drought. Some of this has been put to bed with newer metrics that are slightly more sensitive to, have been shown to be more sensitive to actual photosynthetic productivity. So the idea was to take these limitations and turn them into opportunities. Could we develop a generalized method for mapping phenology that can be applied consistently everywhere? And can we apply that to some of the more sensitive indices of plant productivity that have come out more recently? Things like sun-induced chlorophyll fluorescence. Plants absorb light, most of it goes into food, some of it gets released as heat, a little bit comes back out as light, and we can now sense that from space. And near-infrared reflectance of vegetation, which is a strong proxy of SIF. So these are the two data sets we wound up working with. And I'm going to run through, because there's a lot in the study here, I'm always happy to take questions after. I'm going to run through, as we go through, sort of building out this methods diagram at a pretty high level, but obviously circle back later if you have specific questions. But so where we started was mapping phenology globally. And we did this using both a MODIS surface reflectance data set and OCO2 is another sensor, SIF data set as sort of an independent assessment of the MODIS data. Bunch of quality filtering, aggregation, land cover filtering, and eventually plugging this into a harmonic regression model, which is it's kind of a deceptively naive approach to modeling seasonality. So harmonic regression looks like this. We're predicting, whether it's NIRV or SIF here, as a function of time expressed both linearly and harmonically. So repeating both once a year and twice a year. Anything more complex than that isn't really founded based on the sort of global climatic geography we talked about before. But you can imagine places that would have potentially 2 growing seasons or 2 peaks a year because of bimodal precipitation and so on. And so harmonic regression does this by fitting the coefficients. These are the 4 coefficients of interest on these 4 harmonic components. The sine curve repeating once per year. So everything to the left of this gray line is year 1, year 2, and so on. And the cosine, as well as the sine and cosine repeating twice a year. You take all these things, you fit those coefficients, and a particular set of coefficients on those multiplied together, added up, gives you a model of the characteristic temporal variation within an average year for a given location. And this is what I call a phenocycle. And this thing, the way we model this, repeats every single year over a 20-year time series. So here's what that looks like fitted to real data. Obviously, the black is the real MODIS data and our V data, and the red is the repeating annual peak. And you can see there's variability year to year. That's what you expect. But we're interested in the long-term average behavior of a site. And once we have that, we do some more masking using the Monte Carlo approach to get rid of insignificant pixels. I do comparison between the 2 data sets, again, as a sort of independent assessment, as well as the evaluation of a bunch of ground data from phenology cameras and from flux towers. And then the key here was, rather than melting this down to a sort of what are often used in the literature, sort of univariate metrics, such as start of spring or peak of growing season, instead feeding this into multivariate statistics and empirical orthogonal functions analysis is essentially PCA in a space-time basis. And then taking the top 3 components of that. So the major global kind of components of variability, feeding them into red, green, and blue channels creates a false color visualization, which is common in a lot of remote sensing work, and that gives you this. So this is, to my understanding, the most comprehensive global map of terrestrial phenology. And two things to say about it. One is you pick any two places of interest, the more different they are in color, the more different they are in the shape and timing of the seasonality. The other is if you pick a place of interest, say these peachy colors here, you can compare it to these clusters. This is just the top 9 clusters of this map to give some sort of interpretability, to get some idea of the long-term average phenological behavior. I should have said 3 things. The other things that we fold this across the tropical convergence zone, essentially the climatic equator. And there's unfolded versions and supplements, just to make the opposite hemispheres comparable. So with that in hand, well, so maybe one thing that jumps out here is that there's an incredible amount of spatial variability in the characteristic phenology, right? So with that in hand, we immediately became interested in, well, what exactly makes seasonal phenology vary within regions, even at the local scale as well. We present a whole bunch of mostly both main and supplemental figures that you can look at later and happy to chat more about. But just to show a pair of these, we discover a whole bunch of really complex regional gradients in phenology. So this is the Cape of South Africa. And again, these are just clusters based on this map to give some interpretability. But what you can see here is not only a gradient from essentially the late winter, early spring peaking Mediterranean climate into the summer, mid-late summer peaking continental climates inland, but also that this pocket of perennially wet forest butted up against the massif at the southern edge of the Cape is doing its own thing entirely. And not only are these gradients complex within regions, but they also converge across continents. So this is southwestern Australia and the Cape there. And you get a very similar pattern between the Mediterranean, the gradient from the Mediterranean to continental climates, as well as a pretty distinct thing going on. I believe I've never actually been in Korean Jura forests, it feels like. And that's just one of a number of examples of sort of convergence across regions, across continents. And so this put us in a place where we could actually begin to answer questions about the asynchrony of seasons hypothesis. And the initial question was essentially, well, what exactly does phenological asynchrony look like globally? Does it match, or more or less, the maps of climatic asynchrony mapped by Martin years ago? And one way to look at this is to throw this into a video. So essentially, this is a characteristic year fitted to our 20-year time series. Every day in the year stacked up as a GIF, and every pixel here varies between its own minimum and maximum. Normalizing it in this way draws out the spatiotemporal patterns, right? Regardless of whether we're talking about differences between rainforests with an incredible amount of productivity or arid lands with very little productivity, because I'm not interested in the amount of photosynthesis at the time. So what I encourage you to do is pick a place of interest here and watch it through a single year at a sort of regional scale. I've been calling this my lava lamp because when I do that, especially in regions like the Northern Andes, for example, in places where there's a lot of sort of spatial complexity in the timing of phenology, what jumps out is not only the sort of wave of spring moving up into the Northern Hemisphere and moving back down again, but these really complex patterns at regional scales across individual ranges of the Andes or across divergent ecosystem types in parts of the Amazon and so on. So from this, we could tell right away that there is a really complex global geography of phenological asynchrony, and we essentially developed a metric to actually map that as a static value. The long and short of that is that we calculate a regression for every pixel with all of its neighbors, in which we're predicting the divergence in seasonal timing as a function of distance in geographic space. The steeper that slope, the higher the asynchrony. I can go into the methods more later, but that produces this map here. And what jumps out of this is that there are hotspots of phenological asynchrony in not only tropical mountains, which is what was expected under the original logic behind the asynchrony of seasons hypothesis, but also across all 5 of the global Mediterranean countries. Mediterranean proper, California, also Chile, Cape of South Africa, and southern, even southwestern Australia. And so then we wanted to know what actually drives phenological asynchrony in these regions, and why and how are some why is it what is not only explaining phenological asynchrony within a region, but how does it differ between regions? We put together a big random forest modeling framework for looking into this, modeling phenological asynchrony as a function of a bunch of climate asynchrony maps calculated the same way, as well as topography vegetation structure and some potential confounding groups. And then we mapped the Shapley explanatory values is a sort of machine learning interpretability method that we use to map the predominant drivers that we see. What we found across a whole suite of models is that consistently there are regions where phenological asynchrony across space is driven by asynchrony in precipitation regimes and or minimum temperature. And there are regions where there's sort of codominance, what we call it. And so this is actually mapped as the normalized difference between these top two drivers. And so some of the regions I called your attention to earlier on, like these really kind of stark divisions between winter and summer monsoon regions, we found a really clear signal of precipitation asynchrony driving asynchrony in the land surface phenology there. So you would see something like late winter peaks in Southern California or in the Mediterranean regions of Southern Australia, and then mid to late summer peaks not that far away in the Sonoran Desert or in parts of the outback. And this led to follow-on questions, essentially whether asynchrony between similar climates is higher at lower latitudes. This sounds kind of obtuse, but this is actually crucially important for the evolutionary biogeography piece of this whole thing, because tropical species, especially tropical montane species, have notoriously narrow climatic niches, right, and sort of latitudinal ranges. Altitudinal ranges. And if we expect a tropical species with a very narrow climatic niche to be distributed in multiple sites that are phenologically out of sync and therefore to be diverging evolutionarily, those places would need to be out of sync phenology, even if they're effectively the same climate, right? So say both mid-elevation montane rainforests, and ecologists would walk into these places, call them the same thing, but it turns out the rainy seasons are 4 months effectively. And we develop a clustering approach for essentially clustering individual high asynchrony regions, drawing site pairs within them and asking whether the phenological asynchrony between those site pairs is a function of the climatic difference between the sites, and whether the strength of that relationship varies as a function of latitude. And long story short, we find really strong evidence that there is a sort of general latitudinal gradient in what we call isoclimatic asynchrony. So again, what this means is that at lower latitudes, generally speaking, and there's kind of a more complex geography to this, but at lower latitudes, generally speaking, sites with similar climates are more likely to have asynchronous phenomena. And therefore, even very narrowly distributed species could be distributed in both of the sites and still experiencing essentially allochrony. So finally, this is all well and good. This is all based on remote sensing and a bunch of other giant, global data sets. But the sort of elephant in the room is, okay, does this actually say anything about real ecology on the ground? And so what we wanted to know is, does the land surface phenological asynchrony that we've mapped and kind of explored here predict phenological asynchrony at the level of individual species? We use a whole bunch of different data sets for doing this. Not only all of the flowering taxa on iNaturalist that had enough data for us to look at spatial differences in flowering timing, but also any genomic and genetic data we could find from the few previous studies of the asynchronous seasons hypothesis, and even data from the Colombian coffee growers federation. And what we find is a bunch of kind of compelling evidence that this might be allochrony by allopetra might be affecting a wide variety of taxa. So first, we find that our remote sensing predicts flowering allochrony by allopetra across a whole bunch of species from iNaturalist that are distributed across all of these global asynchrony hotspots that we've found. So we use 7,250 taxa. Again, we put them through a bunch of filtering. They had to have enough data available and so on. About 12% of these have more than one flowering period across their range. And then the question is, well, is it because they're living in the same place, they're flowering twice a year on shoulder seasons, or they're just opportunistic flowering species? Or are they actually flowering at different times in this part of the range than this one? Of those 12%, nearly 1 in 5 showed strong, statistically significant evidence of allochrony biolopatries, actual differences in timing by way of space. And one example here in the Cape of South Africa is a terrestrial orchid. And again, I'll remind you, this is the sort of Mediterranean climate region that sort of swoops down around the Cape. And you can see that the majority of individuals cited in that region have flowers reported in basically late winter, early spring, and the continental climate individuals essentially mid to late summer. And here's a reminder of how that sits on top of the really sharp phenological gradient that we map there. So it's distributed, this holds true for a number of the species we looked at. They're distributed across really strong conformities in our map. We then also found that this actually predicts genetic divergence that's already been attributed to asynchrony, climatic asynchrony in some of the few studies that looked at this before. So the only genomic study, to my knowledge, that has looked at the asynchrony of seasons hypothesis was studying Rhinella granulosa. This is a cane-toad genus relative in eastern Brazil. And this is a region that jumped out in our maps consistently as having really strong asynchrony. And it turns out that just around this break zone in the easternmost part of Brazil, there's a major kind of geographic divide between two completely different sort of precipitation regimes that move in at different times of the year. And if you map all of the author's original sampling points and cluster them genetically, this is what we get. And below are the phenocycles in our map at all those sites clustered the same. If you cluster them instead by our phenology, you get a near perfect match to the genomic data. And not only that, but the only other sympatric species from one of the two only other studies of the asynchrony of seasons hypothesis shows a nearly identical geographic pattern, even though it's obviously very unrelated taxon, suggesting that remote sensing might actually enable us to determine these and describe these kind of largely overlooked by geographic patterns that are affecting a wide variety of taxa. And lastly, sort of sitting under my working hypothesis is that there's knowledge of allochrony biolopatry sort of sitting under our noses that our field has largely just not paid attention to in the past. And one example was literally drinking coffee early in the morning, probably putting up our target on my back here, but I got into brewing my own coffee at home because I got sick of espresso drinks, and Brisbane really loves espresso, and I like espresso, but I also like pour over. Started pouring coffee at home, and then I went down a rabbit hole and became really interested in coffee. And one morning I was literally sitting there thinking, there must be countries around the world that harvest coffee more than once a year, based on our map. And sure enough, I looked that up, and the only country that jumped to mind, and that also came up in an immediate search, was Colombia. And Colombia is known among apparently coffee sort of purchasers and purveyors as having two distinct coffee harvest seasons, fully 6 months out of sync. And Colombia also loves their coffee. Their soccer team are called the cafeteros. It's a big part of Colombian culture, which means that they collect a lot of data on it. And what they call the Fede Café, the National Colombian Coffee Growers Association, has officially mapped 4 harvest season types across the country, varying from basically September to December, to a major harvest season then, but a minor harvest season 6 months out of sync, to a major in the opposite, to a major harvest season, a single harvest season in April to June. And these are mapped here on top of our phenology map in matching colors. And we find extremely strong, significant correlation between these clusters and if we naively cluster our phenology across these sites. And this includes not only this obvious north to south gradient, just to kind of put a point on it, because there is a north-south gradient. This is where the itch passes through the tropical convergence zone. This is sort of the climatic equator, right? We expect things on either side to be the same. But there's not only that, but also this really strong orographic divide where you've got this sort of northern type seasonality comes all the way down the eastern flank of the Andes to such an extent that you've got sites that are only 60 kilometers apart. You could travel between them by a single day's bus ride, and yet they're harvesting coffee as though they're so that's what we cover in our paper. What I'd really be keen to get everyone's sort of thoughts and input and brainstorming on with those I get to meet with the rest of the day today, is sort of future directions for this work. I think there's a number of them, and I kind of break them into two categories. One is sort of generalized mapping and phenology. This is really kind of just the beginning of what I think we could do with this sort of modeling approach for understanding phenology across space. And one thing I think that would be really great to look at is actually starting to understand spatially variable environmental controls in phenology. There's a lot of work on this at regional scales, but I also imagine there are places where you can have very different controls on the long-term average behavior and the short-term anomalies. And annual anomalies are another thing that I think this could be very helpful with. So a lot of phenology literature, a lot of you have probably read some of this literature, looks at things like the advance in spring or delayed, kind of fall leaf senescence and things like that. This is all sort of northern temperate biased, lenses on phenology considering. But we really know very little about arid lands and especially a lot of phenology across a lot of the tropics. And part of the reason is that we can't use those sort of simple univariate metrics in these places. This would allow us to begin to actually map anomalies in a more multivariate and data-driven way and understand phenology in these places, especially under climate change, and get a better sense of how not just the timing of a peak, but the actual overall timing and the shape of phenological behavior is changing under climate change. The other sort of area of future directions here is asynchrony by allopatry. I think there's a lot more work to be done here in the sort of evolutionary implications of this. I think that there's a good chance that the evolutionary effects of this vary a lot across regions and also across taxa within those regions, and we've got a bunch of hypotheses starting to look into that. And we also this doesn't get us to the point of saying, okay, this is actually driving speciation anywhere. And that's a huge question that we need to start pointing at. And it would be good to understand not only if it is involved in speciation, but where and how much and in which taxa. And even whether it's involved in speciation in a parapatric context. There are a lot of places like mangroves and floodplain forests that are characterized by a lot of endemic species and also show really strong divisions phenologically. And phenology is involved in some of the few examples of sympatric speciation, so I think there's a lot of potential there. And I also was talking with someone earlier about this, but I think that it would be very exciting to start looking at a similar approach to understanding spatial variation phenology in freshwater marine systems, especially in the three-dimensional context. And lastly, whether asynchrony by allopatry, the actual landscape patterns of this are shifting under climate change and ecological evolutionary implications. So that's all. We were extremely excited and grateful that we were featured on the cover of Nature a couple months ago. The paper's up the QR code on those left here. And we provide a data viewer so that you can go in and actually run our model live for any 2 points of interest and start running comparisons and looking at behavior of the model and, like I said, poking holes in all of this. And that's at the QR code on the right. Thank you to all of the people and organizations that funded this over the years. And thanks for listening to me rattle on for very today.
All right, we'll open up for questions. We have time for a fair amount of questions, and then I'll let you choose who you want to.
Sure.
And then we'll take a break every now and then to let the people on Zoom ask the question.
That sounds good.
But I'll get us started. Did you include some of the decadal cycles like Enzo or the Indian Ocean dipole in these? Did it have a big effect?
We didn't include them because the modeling framework is so naive that it doesn't explicitly account for anything like these climate oscillations. But it's something that we were very curious about and that reviewers brought up as well. The strong assumption we make is with 20 years of data that gives us enough coverage that it's not hugely influencing our results. But there are places, especially like a lot of Australia, right, that are very strongly influenced by ENSO. And in those places, when you run, like if you were to, for example, go to the data viewer and pick a pair of points in the outback and run the model, you can see huge fluctuation year to year. And those are the places where the R squared in our model is pretty low, meaning that the predictability of the timing of phenology year to year is low, right? Because there's just huge fluctuation interannually because of drivers like that. But the long-term average is still spatially consistent and significant. So short answer is it's not included, but we see a lot of effect of that, and we hope that the sort of time frame kind of partitions that effect out enough that we're still getting accurate long term.
It does make the model predictions less, it reduces your R squared in areas that are highly influenced by these longer term. That's super interesting.
Yeah, like one way of looking at it is that the R squared maps we have in the supplements essentially are sort of an indirect mapping of the interannual predictability of the timing of season.
Cool.
Yeah, thanks very much. It's amazing stuff. The latitudinal gradient in asynchrony wasn't quite as strong as I was expecting. Were you surprised by that?
Yeah, so yeah, when I initially ran the asynchrony mapping, I was kind of naively expecting something similar to the initial Martin et al. Maps I was showing, the climatic asynchrony. And when I looked at the result, I remember distinctly looking at the results and thinking, that's not exactly what I expected. And then looking at it again and thinking, well, it is a lot like what I expected because it hits all of the major tropical montane regions, especially the ones that are more or less perpendicular to prevalent winds, which gets into the topo climate drivers. But it also highlights all of the Mediterranean climate regions, which brought up a whole other kind of round of reading and stuff, because there's some background in California specific and other specific research that describes what's going on there, but it never really been put into a global context.
And do you know what's going on beyond because you're limited in the Arctic, right, by remote sensing?
Yeah, it's the actual, so our reviewers are sort of like, this isn't global, and I was like, well, give me MODIS data for 10 AM and 1 PM in the Arctic, and I'll gladly run it. But yeah, at overpass, it's still nighttime there.
Yeah.
The argument we made was like for the evolutionary, kind of original hypothesis that inspired this. Those were regions where there's very little potential for asynchrony because there's such a short growing season. But yeah, we don't really know, not having the data.
Yeah, that's super cool. And I am not a plant person or an animal person, but just thinking about your first example when you talked about how really strong seasonality at high latitudes, you have this real tight breeding synchrony that you might see them get offset when you move to the lower latitudes. But as well, I think you often see, instead of necessarily like offsetting, it's just like a broadening of that range.
Yeah.
How do you think those two effects, whether it's starting to be asynchronous but still time bound versus just real breadth of that breeding season, what's the ecological impact of those two things being true in the tropics, the different groups?
Yeah, that's an awesome question. I think there's a lot of pieces to this. The one is that that's very much true at the species level, right? But when you look at like the full community, let's say plant or entire community, right, in a site in the tropics, you get a huge range of phenological types, right? Everything from species that are basically just like lots of figs and things are kind of always throwing fruit out, to things that are still very tightly seasonal, and then species that kind of have multiple clutches or are fruiting throughout most of the year. So for individual species, I mean, what I've kind of been saying from the beginning of the study is there are plenty of species that this is not likely to affect, right? But there are still plenty of highly seasonal species in those locations, and I think the coffee example is kind of interesting in this regard, because if you think about coffee as a sort of accidental phenomonitor, I mean, not only is it the same species, but they're often the same handful of cultivars that are highly genetically constrained, just thrown out across a landscape in this really complex phenological landscape, right? It almost serves as a sort of sensor of the driver of seasonal phenology across that landscape, and coffee is highly seasonal still, even in these quote like aseasonal environments, right? And so I think that's an indicator that there are still species that this would be affected by. I don't know if that's answering your question, right? But I think what you'd essentially see is that there are species that this isn't relevant to because their phenologies are never going to be that out of sync between sites because they're breeding throughout the year or whatever. I'm not sure if that answers your question. Yeah, sorry.
Oh, I have a bit of a basic question. How much was your sort of cutoff of data for your iNaturalist flowering example that you included? Like you said,
I don't remember all the details. I think we wound up requiring at least 50 observations. We expected it to be very noisy because it's not iNat, first of all, right? They're annotated by volunteers, they're collected by enthusiasts, they're out hiking and they see a flower. It's not someone saying it started flowering on this date and I'm watching every week. So we wanted well more than just like the 30 minimum. I think we set it to 50 Indian, it's a statistical threshold. But then we also required a bunch of other things. They had to be native species, they had to be not known to be not, like marked as not cultivated, they had to be marked as research grade observations and so on. And then there were a few others we dropped because all of the observations were in the same location, basically, and things like that. But does that
Yeah.
answer your question? But
One
yeah,
more question, I'll follow up later.
okay. So you have your hand up next.
The example you gave for South Africa, because there's orchid, right?
Yes.
It's,
Yeah.
did you say it had 11 different flowering seasons?
No, I mean, there were a whole bunch of observations collected, again, by iNaturalist users, right? And they're out making whatever, they see a flower. And so they're just marked as day of the year, essentially. And we did is we say, okay, we take all those observations and the day of the year of those observations, and we ask, and then we also pull all of our phenocycles at all those locations. We now calculate the pairwise distance matrices between the day of the year as the nearest distance around the year clock, and the Euclidean distance between the shape of the phenocyte. So we've got these pairwise distance matrices that we correlate. But for the visualization, I just clustered them into two because it's pretty obvious that there were sort of two major differences in timing. Yeah.
And that supported your idea of a north-south gradient in richness for South Africa.
Yeah.
The richness gradient there is more east-west.
Yeah, it's not exactly a great, we were not making any assumption about how species richness is distributed or sort of beta diversity. We're essentially saying, is there a statistical signal, a significant signal, sorry, of differences between sites in flowering time that are strongly correlated with differences between sites in remotely sensed land surface phenology. Again, making the assumption that the remote sensing is serving as a strong proxy, at least for some species, of breeding phenology.
Yep.
This might be more of a comment, but I appreciate your view on it, like thinking about marine systems. So on land, temperature is important and light for phenology, but also rainfall. So rainfall is not that relevant in the ocean. But I think the similar driver is nutrients mixing into the upper photic zone, 100 meters, where there's light and phytoplankton plants. And so it's interesting if you look at you were talking about there's different places on land where there's spring and autumn, there's a couple of peaks because of sometimes rainfall in those different times. It's the same in the ocean with nutrients, with spring and autumn blooms.
Okay.
It's just quite a different driver. It's more from wind.
Right.
Leading to nutrients and leading to blooms. And so it would be interesting, I think, that comparison to see, because I think that nutrients and stratification in the ocean is similar to rainfall on land in driving productivity.
Yeah.
So just
Cool. Yeah, that makes a lot of sense. And I'm curious to hear if you agree with my impression has been that at least in some coastal environments, that can be linked to rainfall in places where rainfall is quite varied throughout the year, right? Because a lot of the nutrient inputs require kind of influx of water, right? So is that true?
To 5K or 10K offshore.
Right, okay. Yeah, I would love to chat about this with you more because we put kind of a quick statement about how this might, land on marine and also kind of lodic and lentic freshwater systems in the discussion based on a bit of reading I've done, but that's not my strong suit, and it'd be very fun to brainstorm.
And another comment just relating to comparisons with marine and terrestrial, that lots of, I think maybe this is more marine, but I think the focus has really been on biogeographic shifts, distribution shifts rather than phenological changes. And I think there's more phenological work being done on land because of all the agricultural data sets about flowering, and then also botanic gardens and cherry blossoms, etc. And so in the ocean, there's far more work on distribution change. There's been hardly anything on phenology changes.
Yeah.
So I do think, I don't know, I wondered what you thought about that, whether you've thought about the differences there and in the relative sampling of phenology and distribution, and whether we've been missing something by not focusing enough on phenology change.
Yeah, I'll be honest with you, I haven't thought about that at all, but I would love to, I don't have I don't have background in oceanography. I've read some textbooks for fun, if that's what you can call it, because that's the kind of nerd I am. But yeah, I haven't thought about it much, but I think you're right. There's totally the possibility that you're not only getting one of a number of sort of modes of response to climatic change, right, would be not simply to be shifting in location, but also shifting in location and time, phenologically, right? And to the extent that data doesn't exist, it could be overlooked. Yeah,
I'm sorry. Another interesting direction might be migratory species, right?
Yes.
Because they come out of one seasonal regime and kind of have this, what you're showing is there's this whole smorgasbord of possible seasonal regimes for them to go into at the end of their migration journey. And that strikes me as a kind of a new window on this, because I think people have often thought about what's the broad regions that migratory animals are going to. But what you're saying is this valley versus that valley could be critically different.
And I love the way you framed it too, as a smorgasbord of options, because I completely agree with you. We bring this up a bit in the paper, and I've started some discussions with folks at CSIRO who are working on waterbird migration. I think there's sort of nomadic and semi-nomadic movement, and there's a lot of work around that, but it's not totally clear what's driving that, and we're starting to kind of cook up some ways of kind of getting spatiotemporal understanding of resource anomalies that might drive sporadic nomadic movement. But also, there's like the green wave literature and kind of really well-known tracking of really obvious kind of, like we saw in some of the northern climate parts of our map, right? This really obvious wave. But then there's all sorts of literature documenting. Like I lived in Central America for years, and this bird, the bellbird, will jump between 4 different sites throughout the year, all within like a day's bus ride of each other, and they're predictably there, and it's not really well known why. And there are a lot of kind of altitudinal and transmontane and complex, tropical, semi-tropical movement patterns that I would love to dig into. So okay.
I have a question from Anthony Silverman online. What additional environmental elements feel like the strongest candidates for the next level of analysis here? Are there clear data sets that feel like the strongest candidates you're thinking about that either already exist but need analysis or might be reasonably developed to continue this line of study or inquiry?
Interesting. Yeah. One thing that I've been starting discussions with potential collaborators on is sort of trying to put this into better conversation with kind of ground-based understanding of the cues of phenology, which again are well known in some places in the world, like the northern temperate climates, but can be quite variable between species and are not very well constrained or well understood in a lot of places like the tropics. And so I think using review and some sort of data harmonization or something on the kind of literature locked data on variability in phenological cues across species and across regions and putting that into conversation with remote sensing, I think there's a lot of space there. And then it's not really a new data set, but one thing that I'm really keen to do is to begin to use these sort of long-term average phenocycles as a baseline against which to calculate essentially annualized anomalies, and to start to then bring in other data sets like things about the ENSO index or things about meteorological kind of anomalies, right? Because you could actually start to look at not only the drivers of the long-term average seasonal behavior, but then the drivers of anomalies against that.
Hi, sorry again, did you include any sort of lagged climate? Because obviously it takes some kind of period between the AQ and then phenological response, which in my experience can be quite different, even within the same location. Did you include anything about like a lag response?
We didn't, mainly because you're 100% correct. Mainly because the modeling approach we took kind of obviated that in a way. because the main model, the driver's model I kind of mentioned, is looking at essentially we map asynchrony globally as a metric of a site, right? And then we also map asynchrony in a bunch of climatic variables. And that's based on, I can't remember, I think it's 20 years of climate meteorological data. But we're not actually we're looking at long-term averages, right? But what you're mentioning is exactly what we would need to do if we wanted to start calculating annualized anomalies, sort of year-to-year differences in the sort of fitted phenocycle against the long-term average. You could begin to then calculate that or correlate that not only with, immediate climate, but lagged climate out to a year or multiple years or whatever the case may be in the study system. But there wasn't really space to include it here because we kind of got rid of that by averaging.
Follow-up? Great. So from the little bit of work that I've done in phenology and subtopics, there's at least one species flowering at any month, peaking in its flowering at any month. So there's going to be like this, I guess, squishing effect of the seasonality. So you're going to kind of get like a flat line rather than like a peak in summer or spring.
Yes.
How much do you think that variability may have influenced your global sort of signal? Sorry.
No, don't be sorry. It's a great question. I'm just trying to think of how to respond before I just run my mouth. I don't suspect that influenced the global signal in the sense of like, kind of biasing our results, but that's exactly what I expected to see and did see in a lot of perennifoliar tropical regions, especially where part of the reason that a lot of land surface phenology metrics and approaches struggle there often is that the signal to noise ratio is very low. Just like in arid lands. In arid lands, there's very little signal at all because there's very little greenery. In these places, there's a lot of greenery, but it changes very little throughout the year. So that's the sort of flattening you're talking about, but at the pixel scale rather than the species scale, right? And that's exactly what we see. So there's very little variation in magnitude throughout the year, but there's a very strong, significant signal repeating variation year over our 20-year time span. And it's also, remember, we're fitting each of these pixels independently, but there's really strong spatial coherence in those signals. So even I have a whole bunch of kind of just reserve slides here. So this would be an example, is these are two sites on either side of essentially a range in the Andes. And I mean, there's a bunch of noise here, which is what you'd expect. There's a bunch of dropout because of cloud cover. These are notoriously cloudy regions, right? There's very little variation in the raw and RV values throughout the year. And remember, we put everything through a Monte Carlo analysis and dropped anything that had even a single permuted run with a stronger R squared than the real model. And a bunch of these hold, and all of these pixels independently. I mean, there's some, variation in the sort of tie-dye color, but there's this really strong kind of break across regions like this. And there's this sort of spatial coherence. So we took that as a signal of we're getting something, real, quote unquote. But then at the species level, the strong assumption is that even if we can't from our map predict the date at which a given species is going to flower, but the strong assumption is that between these two sites, if they're say 4 months out of sync in their remotely sensed timing, some species are probably roughly 4 months out of sync in their flowering timing. Regardless of what that timing is, we're basing everything on distance calculations and kind of correlation between distance matrices.
Given that this is sort of going towards the idea that speciation occurs more frequently in the tropics, the world has gone through these ice age cycles over the last 100,000 years or whatever. And do you think this is sort of a snapshot of how these patterns shape up now across the globe, which might have been very different maybe 20,000 years ago? And so how do you see to what extent do you see this snapshot now informative on evolutionary processes which might have happened under, might not, different climates or asynchronies?
Yeah. I love this question, and I think about this a lot too, and I think we're far from a really good answer. If you believe the authors of the Martin et al. Paper, the original asynchrony of seasons hypothesis paper, and there's a bunch of kind of actual mechanistic understanding of the topoclimatic dynamics that would potentially drive a lot of these geographic patterns and so on. They argue that there's every reason to think that similar patterns would have existed over a longer timescale. I tend to agree with that. I think that's likely the case, but I still imagine that everything shifts, right? Everything changes over time. I don't have any reason to believe that any of these mechanisms that drive strong landscape phenology gradients, so say like the way in which the Andes are so high, they block the prevalent trade winds and create the South American jet, which creates this sort of leeward convective circuit that drives totally different precipitation regimes on that side, right? These things are predominantly a function of height of mountain ranges, the direction of prevalent winds, and would have held over at least tens of millions of years, right, in some places. And that would have been more than enough time to drive divergence between populations, presumably, at least for some species. But over the long haul, I don't think we have any understanding. And I imagine that the Mediterranean climate regions, especially because they were subject to such strong glaciation and so on, might have stronger long-term variation in the patterns of allochrotic biolopatry, but I could be completely wrong. Yeah, I think there's a whole lot of questions there and a lot of gray area.
All right, we're running out of time. I'll finish off with the most important question, which is, how do we get this color scheme in R?
You could take a snapshot here and probably feed it into AI.
Did you make it?
The color scheme is just red, green, and blue assigned to the statistical element. So it's sort of determined by our statistics.
But
But
it looks so much better than normal ones.
then what do you mean the
I mean, everything's red, green, and blue. That's not really.
fair enough, yeah. If you take a snapshot of this and put it into ChatGPT and ask for a color scheme that matches it, I bet you get one.
Well, I have your
That's-
paper, but anyway, it's beautiful.
Yeah.
I love
Yeah, thank you. Yeah, it was,
Did you have posters made? I wanna put it up.
no, but I did think about it. It would be kind of fun.
Time.
Okay.
Okay, let's
Look at
give
the
him a
numbers.
big round of applause.